on Sundays, and it's been really fun. So as introduced, I'm Jenny, and I'm currently a postdoc in Ann Osborne's group, which I did my PhD in. And today I will talk about my PhD work, which has recently been published, and the title of Opening a Treasure Trove, Unlocking Saponin Biosynthesis in Soapwort. So, oh, there we go. I guess I don't really have to spend that much time on this slide as we all love plants, and we all know that plants are fascinating, impressive chemists that produce a huge wealth of structurally diverse metabolites that offer protection against biotic and abiotic stresses, but also because these molecules show a wide range of bioactivities. Many serve as important roles in pharmaceutical and nutraceutical industries. Around 50% of the drugs in current use are actually natural products or derived from a natural product. So within these specialized plant metabolites or classes of different specialized metabolites, today I'm going to talk about triterpenoids, which are one of the most structurally diverse class of specialized metabolites with more than 20,000 known structures. And this is just a conservative estimate. And here are some examples of triterpenes that we work in the Osborne group. And these triterpenes are often very heavily glycosylated, and the glycosylated form of triterpenes are known as saponins. And because these saponins are composed of an aglycan core and are decorated by sugars, they have amphiphilic nature. So saponins have soap or detergent-like properties, which makes it foam when you lather with water. So this has been used, which means it has been used as a source of natural soap for centuries. And they also have a very long history of medicinal use as well, with increasing focus on, with increasing focus. Sorry, I didn't realize my thing, but okay. Um, Increasing focus on their pharmaceutical potentials and how they can be enhanced to increase their potency or reduce their toxicity. But because these are plant specialized metabolites, they don't accumulate that high, that accessing these compounds in very large quantities is quite difficult. And they're often in very complex mixtures or very dependent on a particular season or region. So much molecular diversity and their pharmaceutical potentials remain very largely unexploited. So I work on a plant called soapwort, and it's also around me in the background. So soapwort's Latin name is Saponaria officinalis, and it already tells you quite a lot about the plant. So sapo is a Latin word for soap, and officinalis, it comes from the Latin word officina, which means medicinal plant. So indeed, soapwort has been used as such. As a medicinal product for ages. So it is a perennial flowering plant in the Caryophyllaceae, and it's still very widely used in lots of different cosmetics and nutraceuticals and phytomedicine products, with some examples are shown on the slide. And it's very interesting that all part of the plant contain quite high amounts of saponins, which is also have important pharmaceutical properties, such as being anti-cancer or used in drug delivery. So here, I'm just going to get my pointer out. Here, I just have water, and this is my water control. And here I just sort of like put some flowers in it and some ripped leaf in it. I just shook the vial for about 10 seconds. And you can see that there's this very nice layer of foam that's visible in the flower and the leaf sample, but not in the water. And I hear that, so beer industry actually uses soapwort extracts or saponins to make this nice beer head, which apparently I don't really drink, but apparently it's nice to have foam in your beer, which I don't really understand because I feel like you should get more beer than foam, but I'm not drinking it, so what do I know? So here is a picture of soapwort. So within the saponins in soapwort, one of the major saponins that are reported from soapwort is this compound saponaris A and B. So these have a oleic acid aglycon, and then they are decorated at the C3 and the C28 sugars with C C28 positions with two sugar chains, and they're quite highly decorated. At the C3, we have glucuronic acid, xylose, galactose. At the C28, we have arabinose, fucose, quinavose, which I'll talk about later on, but they're quite unusual sugars in plant specialized metabolism with an acetyl group, and then there's also two xyloses there. So it's quite a big molecule. And saponin A is only different between the two compounds is just the addition of a xylose here. So saponin A has an additional xylose, while B does not. And this highlighted part actually represents a common saponin scaffold that's found across the Caryophyllaceae. And perhaps more interestingly, this compound is also found in, well, very similar in structure to QS21, which is a saponin found in the Chilean soapbark tree, Saponaria officinalis. Despite the two plants being quite phylogenetically distant, soapwort is in the Caryophyllaceae order, while soapbark is in the Fabalius order. So QS21 has been very extensively investigated for its immunostimulatory properties, and the full biosynthetic pathway has been recently elucidated by our group. And QS21 currently is used as a vaccine adjuvant in vaccines such as shingles, malaria, as well as Novavax's SARS-CoV-19. So despite although the similar structure doesn't mean that it might have vaccine adjuvants, but there are quite a lot of pharmaceutical potentials to saponaricides. But despite this, there has nothing been known about its biosynthetic pathway. So that's sort of when I started this project. So beginning of the project, there wasn't any much publicly available sequence data except for a single transcriptome from the 1KP project. And we wanted a more comprehensive sequence resource. So we generated a pseudochromosome genome assembly together with our collaborators in the States. And the genome, I said that I was going to change this, but it's not. It should be moved here. Or maybe I do have it right. Anyways, it's 2 gigabytes. So it's quite a big genome. And then after I started to look at more in detail in the metabolite accumulation in the plant, specifically on saponin A and B, to see if there's any kind of tissue-specific accumulation. So I divided the plant into 6 different parts, and I specifically looked at saponin A and B, and you can see that there is this differential accumulation of A, mainly accumulating the flowers and in the roots, and saponin B is more dominant in the green tissue. Regardless, this differential accumulation pattern gave us this opportunity to generate tissue-specific expression data by RNA-seq. So with the newly generated sequence resources, we then started to search for the genes involved in saponin biosynthesis. So saponin aglycone is a cholic acid, which is derived from beta-amarin, and the first step of triterpene biosynthesis in plants all begins with this universal triterpene precursor of 2,3-oxydosqualene and the cyclization of this 2,3-oxydosqualene by an oxydosqualene cyclase to the various triterpene backbone, which indicates I was looking for a beta-amarin synthase. So this was identified through phylogenetic analysis with known OSCs from other plant species, and there was really one that was seemed like an excellent candidate. And in our group, we like to test our functioning genes through heterologous expression in Nicotiana bentamiana, and it's an absolutely brilliant system. You can scale it up to however you want. You clone your expression vector of interest, transform your agrobacteria, and infiltrate the tobacco leaves. And after 5 days, you just extract and analyze to see if your compound of interest is there. And this can be scaled up quite a lot to like 100 to 200, 300 plants. And you can produce gram quantities, up to gram quantities of pure compounds. So I did this just small scale to test out the activity of this potential candidate. And indeed, if we were to infiltrate the tobacco leaves with SO-BOS1, which I named the candidate as, it was producing the same peak that was corresponding to a beta amarin standard. So this step was solved. So using that first gene that we just characterized, we then started to mine our sequence resource for further biostatic genes using co-expression analysis by using the beta amyrin synthase from soapwort as a bait gene. And then we also looked at other things such as the absolute the transcript number in the flowers, because just to remind you guys, it was in the combined accumulation of saponins A and B was quite abundant in the flowers and the flower buds. And we thought that perhaps this means that there's very likely chance that perhaps the compound is biosynthesized as well. Then it would mean that the transcript levels would be quite high. The expression would be high in the flowers and the flower buds. So we were looking at the transcript read counts in the flowers. And then we also filtered it by relevant biosynthetic families, such as cytochrome P450s, UDP glycosyl transferases, and acyl transferases, and such. So after getting this table of candidate genes, then we heteroglyphically express these candidate genes in different combination in equation of Pantamiana, and then analyze the leaf extracts through gas chromatography or liquid chromatography, LMS analysis, and then we compared the product peaks with authentic standards. So there weren't a lot of very little later on in the pathway. There weren't a lot of standards that we could commercially buy, so we had to generate a couple of them ourselves. So after rounds and rounds of different heterologous expression experiment, we were able to complete the biosynthetic pathway to saponin site B, which involved 14 pathway enzymes. And as Lars mentioned, that this is recently published here. And call me biased, I think it's a great read, so please do take a look when you have free time. So the nitty-gritty bits are all in the paper, so I won't bore you to details by going through the entire pathway. But first, I'll go through some bits that I find quite exciting, which is this bit where we have the quillic acid backbone. So from oxidosqualene, we made our quillic acid backbone, and then we've added a glucuronic acid, a galactose, and a xylose, and then we are here. And the next sugar to add on the C28 is this D-fucose. And I'm just going to highlight a little bit here because D-fucose itself is quite interesting as it's not made at the sugar nucleotide level. D-fucose is sort of an unusual deoxyhexose sugar, and there's no reports of corresponding sugar nucleotide in the plants. And its biosynthetic pathway, or how D-fucose is made in plant, has been elucidated by our group when they were looking for the QS7. And this was really great because that means that I could also sort of have this as a guideline to go through as well. So what this paper found in our group is that D-fucose is actually formed from 4-keto-6-deoxyglucose, which is an intermediate of a UDPL rhamnose biosynthesis. And a UGT actually attaches a 4-keto-6-deoxyglucose to the saponin backbone, and then a short-chain dehydrogenase reductase, in this case in Chiara, called chiara-fuchsine, actually reduces the 4-keto-6-deoxyglucose to fucose. So with that guideline, we also looked for a potential short SDR in soapwort as well. And I'll just walk you through this chromatogram. So this is LCMS chromatograms extracted ion at 1101, which is the mass of this compound, which is a shared intermediate between the QS21 pathway as well as my saponin side pathway. And so I've infiltrated all the genes required to get to cholic acid backbone with 3 sugars at the C3 position. And here I added a candidate SDR from soapwort, and we don't see any accumulation of product peak. And here I've just added UGT to the mix of all the genes required to make Q8 tri, and we see a formation of 2 peaks, well, 3 peaks, I guess, but this is the peak that corresponds to our compound of interest that we want. And then it's only when we add the UGT as well as the SDR, we see this increased boost of the saponin compound with the additional fucose. So the addition of fucose is in soapwort follows the similar mechanism as to Chiara. So after the addition of the fucose, we have an addition of a rhamnose, two xyloses by UGTs. And then the next part that I think is quite interesting is the addition of another quite unique sugar, which is quinavose, to the saponin backbone here. So this is catalyzed by this gene SOGH1. And SOGH1 is a member of the carbohydrate active enzyme like UGTs, but they're quite different. So it belongs to the glycosyl hydrolase 1 family. Instead of UDP sugars, it uses acyl sugars. And all characterized GH1 TGs so far are targeted to the vacuole. And there are a few that has been reported with roles in specialized metabolism, mainly involved in anthocyanin biosynthesis and the transfer of glucose. So D-quinavose, as I sort of hinted, is also a rare sugar, an unusual sugar in plant specialized metabolism, and its biosynthesis is not yet known. But interestingly, if I were to add all the soapwort genes required to make this precursor saponin, and this gene SOGH1, which we found through co-expression analysis using the SOBOS1, the first step in triterpene biosynthesis as a bait gene, we get a product with an addition of D-quinavose in it. And I guess I should have shown these first. So yeah, we get 2 products where this product matches up to our authentic standard of this. And this is just MSMS spectra, just showing you the fragmentation pattern. So this molecule has a mass of 1657. When we do tandem mass spectrometry, we get a daughter ion at this compound, which is Q8 tri, so cholic acid with the 3 sugars on, which corresponds to a mass of 955. And you might wonder what this other peak is as well. We think it could be just a xylose. So instead of xylose being attached here, perhaps it's getting attached to a quinavose but this is a putative identification as well as these 2 peaks share the same fragmentation pattern as from 1657. The main fragment ion is this 955 peak, which is the mass of cholic acid with the 3 sugars attached to the C3 sugar. So this is to do in my to-do list to purify and to get the structure of this as well. So the result of SOGH1 was quite surprising because to me, I didn't really, when I was first starting the project, I knew that quinavose was going to be a problem because one, there's really nothing reported on the biosynthesis of quinavose. And I actually didn't know what quinavose was when I started. So it was a very pleasant surprise when we found this gene through co-expression and it produced what we wanted. So then we decided to investigate SOGH1 a little bit more farther. So glycosyl hydrolase TGs normally have an N-terminal signal peptide. So we performed signal P analysis on characterized GH1 TGs, which are shown in bold here, and some predicted ones in soapwort, including our GH1. and other plant species. And signal P basically just detects the presence or an absence of a signal peptide on an amino acid sequence. So in this heat map, the greener it is, it's more likely to have a signal peptide. For example, GH1 TG from Avena, from oat, we know that it is in the vacuole, and you can see that it's quite green. But if you take a look at soapwort, it's quite black. Which was quite surprising. And when we actually did look at the amino acid alignment with characterize GH1 TGs together with our SOGH1 from soapwort, we see that it is missing this signal peptide from in the N terminal that's highlighted in green in the other plant species. So then we, of course, decided to take SOGH1 first with red fluorescent protein, and then to really look at its localization. And indeed, it was a cytosolic enzyme. Um, so this would be the first to be reported in the GH1 TG family to be localizing to the cytosol. And we also looked at the activity as well. The tagged protein had the same activity as the untagged protein. So now we are sort of attempting to resolve the substrate and donor specificity of SOGH1 as well as quinavous biosynthesis in plants. And we sort of started to sort of touch on it. To kind of figure out what kind of what kind of donors GH1 used. So this was done by Shingo, a postdoc in our group. He was able to purify the enzyme really beautifully and did a lot of in vitro assays. So this was a surrogate saponin backbone that we were using as the actual precursor before the SOGH1 step. We couldn't get enough saponin, but we had this in our lab. The only difference is that instead of this rhamnose in the C3 chain, I have soapwort has a xylose. And unfortunately, it'd be really great if we could get acyl quinavous commercially available, but we can't buy anything like that. So we sort of looked through anything commercially acyl sugar wise commercially available, and we sort of opted for different acyl glucoses and also different things that we got our hands on. And we did some in vitro assay, and it seemed like it was able to SOGH1 was utilizing an acyl sugar as a sugar donor, which is in line with the characteristics of GH1 TGs as well. And just a little bit note of quinavous biosynthesis as well. So I mentioned D-fucose before, and D-fucose and quinavous are actually C4 epimers. So this is 4-keto-6-deoxy glucose, which is the precursor to getting to a D-fucose. And here you can see that quinavous and fucose are just C4 epimers. So our current working hypothesis is that the quinavous biosynthesis may be very similar in mechanism to the fucose biosynthesis as well. We think that it will start with UDP 4-keto-6-deoxyglucose, and perhaps an unknown UGT will put it to an acyl backbone to make an acyl 4-keto-6-deoxy-D-glucose. Here we just have benzoyl benzoyl for illustrative purposes, but we don't know what the acyl group is going to be like. And then we have two routes here. One is that a unknown 4-keto reductase reduces this into quinavous, or SOGH1 takes the 4-keto-6-deoxyglucose and puts it to a saponin backbone, or SOGH1 works with the acyl quinavous as a donor and put it to the sugar backbone. So there's a lot to figure out here, and this is sort of what my postdoc work has been for the last couple of years. So after that step, I guess back to the pathway is an acetylation step catalyzed by a SOBHD1. And when we infiltrate all the genes up to here with an addition of SOBHD1, we get a product formation of two peaks again. And the first peak corresponds to our saponin B standard. And then the second peak, we didn't really know what it was, so we had to purify it. Actually, this peak is actually quite abundant in the flower, so we were able to purify it from soapwort flowers and get NMR data on it. And it turns out that it's this structure isomer to saponin B, which I termed it as SO1699. So saponin B has a xylose in this chain, while saponin SO69 has the xylose is being attached to the quinobus moiety here. So with that, saponin B is one step away from saponin A. We still require an additional xylose here to get to saponin A. And it's quite interesting as SO6999 could also be a precursor to saponin A, but it could be a precursor to this other saponin that's highly abundant in soapwort called SO1861, which is also called sapofactocid. So this molecule is reported to augment the toxicity of type 1 ribosome inactivating proteins, which are also present in soapwort, and they're called saporins. So this is really fun because saponins, saponariaceae, saponaria, and then we have saporins in the mix. So the saporin, the type 1 ribosome inactivating protein in soapwort, is highly investigated for targeted therapies against Alzheimer's, epilepsy, cancer, and such. So with the complete elucidation and the reconstitution of saponin B biosynthesis, we now unlock new avenues for accessing and engineering valuable saponins, such as I mentioned, SO1861, which is a drug delivery enhancer. And saponin B, which we have shown potent anti-cancer activity. And of course, we also present a unique set of enzymes to produce QS21 and its analogs. As enzymes of saponin biosynthesis and QS21 biosynthesis with the corresponding enzyme activity, they share quite low amino acid similarity, except for the very first two steps. And I'm just going to spend a little bit time on last few slides on why my title is called Unlocking This Treasure Trove. Or so, as I mentioned before, saponin there's a lot of saponins with similar structures to saponin that are found widespread in the Caryophyllaceae family and across the Caryophyllales. So here we have saponin B, and you can see that there is very structurally similar saponins all around in the Caryophyllales. So potentially, we can use saponin biosynthetic genes to elucidate the pathways through orthogroup analysis. And we started to go into this a little, and you can already you can already start identifying candidate genes that might be involved in saponin biosynthesis in these individuals here. In the Caryophyllaceae, it's quite interesting as we can find all homologs in different Caryophyllaceae species. But once we move away from the Caryophyllaceae family and to Amaranthacea and Polygonacea, there's less and less homologous genes found here. So this really opens up this opportunity to mix and match different enzymes from different species and to start designing complex molecules and novel saponins with potential pharmaceutical applications. And then we can also start to investigate the evolution of saponin biosynthesis in the Caryophyllaceae order more broadly by investigating the evolutionary relationship between the enzymes discovered from soapwort and potentially in the closely related species and the less closely related species or families in the Caryophyllaceae members. So with that, I'd like to thank, of course, and for her support and the project and everybody in the group as well. So Amr did a lot of the different purification. He's now at UEA with his own group. He was really great purifying all the sticky saponins and also the various JIC platforms and our collaborators. And again, thank you so much for inviting me. And thank you very much for your attention. Should I stop sharing?
Yeah, you can stop sharing. Thank you very much. That was a great presentation. And if there are any questions, please feel free to raise your hands or put your questions in the chat. Yeah, we have one question. Please go ahead, Jack.
Hey, Jen. Yeah, nice presentation. I have two questions. The first one is, in your first distribute bar graph of the distribution of the SAP A and B, you show that SAP B is also accumulating roots. Um, I wonder how co-expressed are these genes, and then do you think there's a transport mechanism for SAP A from the flowers to other parts of the plants?
That's sort of a question that I have as well. It's either there's two different, because these genes are very highly expressed in the flowers and they're very a little expressed in the roots, not very much. So it could be that there's a different set of genes in the roots that is very specific to saponin side A, which I don't think it's very likely. So perhaps there's likely to be a transport system between the flowers and the roots, but I haven't looked at any potential transporters yet, but that has been in my mind as well.
Okay. My second question is, we kind of present this pathway as to build up the sugar on the left hand side first, and then the right hand side. But do you think is this like a linear pathway, or it could be more, it's not that older?
Oh, sorry. Yeah, that's a good question. If the C28 sugar could be first, then the C3 could be first, right? So
Right.
I tested this as well. I think this, because my project was sort of done similarly to the QS21 project, and in the QS7 pathway, they really do need to put the C3 sugars first. And when I tried it with my soapwort genes, I got very, very low result when it didn't have the finished C3 sugar chain. So I think it does need the C3 sugar chain, and then it goes to the C28 sugar chain. As the fucose transferase, so the first sugar at the C28 is the fucose, and it just would not attach without sugars on the C3.
Okay. Yeah, that makes sense. Thank you.
Thanks, Jack.
Yeah, Gaurav, please.
Hi, nice talk. So I had a question about the fucose synthase. So there are fucose transferases too, and I was wondering the GDP fucose transferases. Uh, and I was wondering if there is what made you think of the synthase mechanism over a transferase mechanism, or if you found any transferases too in in the data set.
Um, Sorry if just double
Okay.
checking the question. Is it on the mechanism of fucose biosynthesis and transporters related to that?
The fucosal transferases, like
Sorry,
the
could
I'll type it in the chat. Fucosal.
Oh, I see. Yeah, so this is work that was done by or published in the science paper with the Chiara and fucose biosynthesis. So it's actually it's not actually a fucose transferase. It's a UGT that actually puts 4-keto 6-deoxyglucose onto the saponin backbone first, and then a short chain dehydrogenase reduces the 4,6-keto deoxyglucose into fucose. So the
Right,
UGT
right,
doesn't transfer fucose at all because UDP fucose just hasn't been found or reported in plants.
right. But I thought there were GDP fucosal transferases in plants. GDP, not UDP, but
Oh,
GDP
GDP.
fucosal trans.
I thought GDP was specific to microbes. Is it
I
in
could be
plants as well?
mistaken. I could be mistaken,
Okay.
but yeah.
I'm not
Uh,
sure about that part. As I said, the fucose part was sort of done by the the Kyohara team as well.
right. Yeah, because we have EC fucoses too in our metabolites, and we were wondering how those are added. And in our data sets, we sometimes see fucosyl transferase containing domain proteins that are seen in our data set. So that made me, this was very interesting. I'll definitely go check your paper. My second question was, do you know about the localization of these syntheses? You mentioned the glycosyl hydrolase is in cytoplasm, but where does the glucose to glucose to fucose occur?
Usually, UGTs are cytosolic proteins. So when we were touching, so I'm not entirely sure where the whole fucose and quinolvose biosynthesis is happening in the localization. But that is it is of interest as well. That's sort of what I want to look at as well, where these where the biosynthetic pathway is actually happening in the plant. Because when we're testing out the BAH, the next step is the BAH deacyltransferases, and those are known to be cytosolic. And the step before the GH is also a UGT, which are likely to be cytosolic proteins. So I feel like perhaps it's being produced in the cytosol and then gets transported into likely to be a vacuole for storage, likely. But we need to figure it out.
Thank you.
Thanks.
Yeah, Luke, please.
Hi, Jenny. Thanks for a great talk. Just sort of a quick question. In your transcriptome data, and I guess maybe this might be difficult to answer based on how the assemblies were, but did you see a high number of closely related homologs for many of the downstream pathway genes, like not the OSC maybe or the P450, but were those, it seems like you found some good candidates pretty quickly based on co-expression and expression levels in the relevant tissues. But I'm wondering about the sizes of those, if you have any information on that.
That's a good question. I'm trying to think when I was looking at my sequence data. I think sometimes there were, but it wasn't a problem. Like it wasn't something I had a lot of multiple very similar genes. There was maybe like one or two that might likely to be like a different assembly, but then we also had the genome to go back to as well. So that was more concrete. But yeah, there was one or two that might be very similar to each other, but I think it wasn't a problem when I was cloning out or looking at the sequences itself.
Amazing. Thank you.
Thanks.
I also do have a question. I was wondering how much problems did you have with like unspecific side products when you were trying to reconstitute the pathway in Nicotiana bentaminiana? Because all these sugars, I imagine that there's lots of unspecific glycosylation as well.
Yeah, I think that's probably likely because as you build up the pathway, right, you see lower yield. And that's probably because of this untargeted modification that's happening. If I'm being honest, I sort of turned a little bit of a blind eye here, and I looked at very specific target masses as well. But for most of my enzymes, the accumulation was quite good. There was very little obvious off-target activity as well. But that is also to say, like, I wouldn't say that there is no off-target activity as well.
Yeah.
But it wasn't much of a problem unless you get to the very end. But then again, it's like, is it because I infiltrated so many genes or yeah.
Yeah, thank you. Yes, please.
Oh, hi, Jenny. Very nice talk. I
Thanks.
have two questions. So one is, are there any other reports that the glycosyl hydrolase is involved in secondary metabolite biosynthesis? And my second question is like how many genes, like maximum number of genes you infiltrated into tobacco and got a product?
So the first part, oh my God, I forgot to know already. I had an answer in mind and I just forgot.
Sorry, I
Could
can
you?
repeat. Yeah, so the glycosyl
Yes.
hydrolase. how
Yeah, I
frequent
got
do
it.
you see it?
So yes, there's about like couple of there aren't a lot that's identified so far, but there has been about a handful that's identified. So for example, there's glycosyl hydrolase transglucosidase that's involved in adenosine biosynthesis, as well as there's quite a lot in, well, about 5 or 6 in anthocyanin biosynthesis as well. But all of them are known to transfer glucose. So quinobos is sort of like a new thing, and they're also all targeted or sequence-wise to be predicted to target to the vacuole as well. And the second question, I've forgotten already. I'm so sorry.
Don't worry. What's the maximum number of genes you express in tobacco and saw the expected product?
So me personally, I think it's 30. So when I was looking for the glycosyl hydrolase, this was actually when I was looking for all the different GTs. So I think I had like 16 GT candidates and then my pathway genes, and then I put everything in there. And you actually like see a good product, which is really surprising. But that's me personally. In our group, I think the record was like 60 or something. So you can really go crazy with this and you do get something out of it. So it's quite impressive.
Thank you.
All right. If we don't have any further questions, I would like to thank Jenny again. Wonderful presentation, very nice discussion. I'm very happy to have you.
Thank you again for the invitation.